Good afternoon, everyone. First of all, it's wonderful to see such a robust turnout for today's lecture, and so it's a pleasure to welcome you here. This is the 2024 Alan S. Michaels Lectureship in Medical and Biological Engineering. This lectureship was initiated in 1995, almost 30 years ago, and it has served as a forum for distinguished engineers and scientists in academia and in industry to share their views on research and development in biomedical engineering and bioengineering. Before I introduce today's speaker, I'd like to say a few words about Alan Michaels. Alan Michaels was a very important part of both our department and of MIT's life beginning in the 1940s, when he received his SB in 1944, the MSc degree in 1947, and the PsyD degree in 1948, all in chemical engineering. Alan subsequently joined the MIT faculty and worked with Warren K. Doc Lewis and Ernst Hauser in the teaching of colloid chemistry. After a year in industry, in 1950, he returned to MIT to assume the position as co-director of the Soil Stabilization Laboratory, which at that time was a new joint effort between the Departments of Chemical and Civil Engineering. Dr. Michaels' research interests at MIT were innovative and diverse, including colloid phenomena, soil mechanics, and polymer permeability, and its dependence upon polymer structure. He also had a particular interest in large impact and the promise of polyelectrolyte complexes and coacervates. His efforts pioneered the development of perm-selective polymeric membranes, which led to the great interest in membrane separations that continues today for both gaseous and liquid mixtures in the chemical process industry. In 1962, he founded Amicon Corporation, which is now a part of Merck Millipore, which produced filtration materials for separations processes used by the biotechnology and healthcare industries. He left MIT in 1966 to serve as Amicon's president. Alan's many honors and awards include election as a founding fellow of the American Institute of Medical and Biological Engineering, a society in which our guest today is also inducted. In addition, he held several distinguished academic appointments, as well as industrial and scientific advisory positions for the food, pharmaceutical, and chemical industries. He was elected to the National Academy of Engineering in 1979 for, quote, pioneering developments in the fields of surface, colloid, and polymer chemistry, membrane science and technology, and advanced systems for drug delivery to the human body. Alan Michaels was a pioneer in the application of chemical engineering principles to bioengineering. His leadership in forging cross-disciplinary collaboration between the medical and life sciences and chemical engineering has been recognized by the creation of the Alan S. Michaels Distinguished Lectureship in Medical and Biological Engineering. And now it's my pleasure to introduce this year's Michaels lecturer, Professor Jennifer Lewis. Professor Lewis is Jianming Yu Professor of Arts and Sciences, the Vice Professor for Biologically Inspired Engineering in the Paulson School of Engineering and Applied Sciences, and a core faculty member of the Wyss Institute at Harvard University. She received her bachelor's in ceramic engineering from the University of Illinois Urbana-Champaign and her PsyD in ceramic science from MIT in 1991, conducting her doctoral research under the supervision of Professor Michael Sema, and she was his first graduate student, I learned this morning. Professor Lewis began her academic career at her undergraduate alma mater, and she joined the faculty of the Harvard School of Engineering of Applied Sciences in 2013, where her research focuses on biomanufacturing vascularized human organoids and tissues for drug testing, disease modeling, and therapeutic use. In the vein of Alan Michaels, Professor Lewis's work on 3D printing has had a strong focus on translation and commercialization. She's co-founded several companies based on core IP from her lab, and 3 others are commercializing technology in 3D bioprinting, also licensed from the Lewis Lab. The applications of these ventures range from drug delivery to kidney therapeutics. Professor Lewis has received numerous awards for her work, including recognition as a Fellow of the American Physical Society and the Materials Research Society, and is a Vannevar Bush National Security Science and Engineering Faculty Fellow. I will say personally, I am thrilled to see that she was a recipient of the Lush Prize for Scientific Research. You can see it there. I have 2 daughters who will know that I have met a rock star today because she was recognized by Lush for work towards ending or replacing animal testing. She's an elected member of the National Academy of Sciences, the National Academy of Engineering, the National Academy of Inventors, and the American Academy of Arts and Sciences. Please join me in welcoming and extending a very warm welcome to this year's Alan Michaels Lecturer.
Thank you. Very kind. So let's switch. All right. Thank you so much.
Oops.
All right. I'll let you pull that up.
I'll get back to yours.
Well, while you're switching that, let me just begin by thanking the department, thanking the Michaels family, and particularly Stephen. So glad that you could be here today. For this wonderful opportunity, and for Paula for extending the invitation, and the department for being such gracious hosts today. Really have enjoyed meeting the young faculty, the students and postdocs that I had lunch with. It's been quite a wonderful experience so far. So today I'm going to share one kind of deep story about our work in biomanufacturing of human tissues for drug testing, disease modeling, and therapeutic use. Since joining Harvard roughly 10 years ago, our lab has embarked on drinking from the fire hose of biology, learning a lot about stem cells and organoid differentiation, and trying to use our core expertise in direct and embedded bioprinting to kind of combine top-down and bottom-up strategies for building vascularized tissues. We've employed techniques like flow-enhanced vascularization to upregulate and mature organoids, and we've used these organoids both in the drug testing and disease modeling space, but also as building blocks for organ-scale tissues. And we've also worked in the cerebral tissue space using orthogonally induced differentiation. And I know, oftentimes in a lecture like this, people will highlight multiple threads of work that they've done. But today, I'm going to really, as I said, kind of focus in on our kidney work, starting with examples of using organoids for drug testing and disease modeling, and then describing our efforts to really scale up these as building blocks for vascularized tissues. Now, one of the reasons why we have focused on these kidney-on-chip models is because there's a tremendous amount of money spent every year to bring drugs to society, and that money goes to waste because drugs fail, often in late stages of the phase 3 clinical trials. And so I think the field in general is embracing this idea of having personalized medicine platforms, having platforms that are like fail fast, phase 0, using human tissues with the complexity to really model and mimic the in vivo tissue environment. And just to point out, 20% of these new drugs fail due to nephrotoxicity. So they don't make it out of clinical trials because they're toxic to the nephrons, which are the key filtration units in our kidneys. And of course, from the larger or longer term perspective, we're also very interested in therapeutic use of these kinds of tissues that we're engineering and building in our lab. And if you look at the number of people waiting throughout the world, the donor on the donor wait list, the organ of most need is a kidney. So in the US alone, there's roughly 100,000 people waiting for a donor organ, and throughout the world, it's on the order of almost 1 million, right? And while there is a bridge to getting a kidney transplant, known as hemodialysis, that is actually a disease in and of itself. Once you go on hemodialysis, every year your life expectancy reduces by another 15%. So that's not a really great option. And even if you are so lucky to get a donor organ, you have to be on immunosuppressants the rest of your life, right? So, our wish and our hope is to build autologous tissues made from you for you to address this important need. And if we do that, we would reduce rejection and also alleviate this growing, ever-increasing gap between the donor supply and demand. So let me tell you a little bit about the kidney and how it works. We each have two kidneys in our body. They are responsible for removing waste from our blood. Once blood goes into the kidney, what comes out is filtered blood, and what is removed as waste in your urine. And each of our kidneys contains a filtration unit known as the nephron. We have about 1 million filtration units in each of our kidneys. So they're working in parallel to do the cleansing of our blood. And they contain many compartments of interest. So first of all, in the glomerular compartment is where the blood flows in, and I'll show you in a little bit the slit diaphragms that are responsible for the filtration. The filtrate moves through the proximal tubule compartment down into the distal tubule, and then ultimately is excreted as urine. But throughout these tubular compartments, which are wrapped with capillaries, we have reabsorption. So my postdoc that really started this project likens our kidney to like telling your kid to clean his bedroom, and if you come to the bedroom and everything has been moved out, not just the dirty clothes and the bed's not straightened up. So in this part of the nephron, the good stuff goes back into the bloodstream. All right, so we're reabsorbing nutrients and things. And it's also responsible for homeostasis, for maintaining homeostasis. So now if we zoom into the kidney over this multiple length scale, like I'm a material scientist, right? So I like to think about building blocks from the molecular scale to the macro scale. We can also think about the kidney from the macro scale down to the sort of nanoscale. And if we look then into the glomerular compartment where the capillaries are packed in these little tight clusters, and we look at the podocytes, which are the cells that have these little foot processes that extend, that basically serve as the filtration units that wrap around the capillaries. And then these are nested in and connected to the proximal tubules, which is where all the reabsorption occurs. So if we think about engineering kidney tissues, we really need to be able to recapitulate not only these architectures, which are responsible for function, but also the myriad of cell types, the podocytes, the proximal tubule epithelium, the distal tubule cells, et cetera, and arrange them, as I said, in this exquisite architecture. And then throughout this, we need to embed vasculature. Not only do we need to sustain the cells, and the vascular supply provides the oxygen, the nutrients that do so, but of course the whole entire process is blood in and blood out, right? So this vascularization is quite important. So we started this project thinking, really we started working on engineering kidneys about 10 years ago, and fortunately around this time, there was a major advance in being able to take patient-specific cells, adult cells, and reprogram them into the induced pluripotent state. And around the same time, there were also myriad protocols coming out to take these aggregates and differentiate them into organoids. They were referred to commonly as mini organs or organs in a dish, largely for pharmaceutical, for drug screening. But we recognize that beyond that, they could also be very valuable as building blocks for tissue engineering. So with that in mind, the stories or the work that I'd like to describe today is really divided into sort of two sections. One, looking at how we can build kidney organoid-on-chip models and use them for drug testing and disease modeling. And the second one is a process that we define as SWIFT, sacrificial writing in functional tissue, where we take large assemblies of these individual organ building blocks, compact them, and use top-down printing simply to write the vascular network. So we really want to, unite, if you will, stem cell biology with bioprinting to create these kinds of architectures. So let me begin by describing some of the work that we've done on vascularized organoids on chip. And I want to maybe begin by highlighting that one of the real bottlenecks when we started working on this was that you could make these organoids, but they didn't typically have a vasculature. And that's true for cerebral organoids, for other types of organoids as well. So being able to actually introduce microvessels at the scale and integrate this in the organoid building blocks themselves had not yet been demonstrated. So we had been working on just simple proximal tubule on chip models, and we realized that flow was central to enhancing the morphology and function of these cells. And we posited that maybe flow would be very important in upregulating the maturation of organoids. And that really is how we started that work. And then more recently, we've taken this model and we've introduced an immune system so we can probe things like T-cell bispecific antibodies and interactions with the immune system. And then we've also partnered with Ryuji Morizani at MGH to work on polycystic kidney disease, so to take CRISPR-Cas9, create disease models. Today, I'm not going to highlight this work in any detail, but I will say that it was actually a very promising outcome from that work. We identified 2 FDA drugs that could be repurposed to help autosomal polycystic kidney disease. So one of the drivers for developing these models, of course, is very recently the FDA passed the Modernization 2.0 Act, which now says that pharmaceutical companies can go directly to clinic with preclinical data based on human models alone. And so there's a real need, of course, to develop these kinds of human models that recapitulate organ-level microstructures and cellular architectures. And just to give you a sense, we're working closely with Roche Pharmaceutical. They've recently invested and stood up an Institute for Human Biology. It's roughly 120 people now, but their goal is to build to 250, and it's all based around organoid-based research and somewhat on organ-on-chip level, some more simple models, with the idea of using these and developing these for their internal drug targets and drug discovery programs. So why organoids, and why combine those with organ-on-chip models? For those of you who are aware of the organ-on-chip work from Don Ingbar out of the Wyss Institute, those are typically porous polymer membranes that are seeded with cell monolayers on both sides of the porous membrane. And they do have flow. So you have multiple cell types. You don't recapitulate the three-dimensional architecture, but you can have human cells and you do have flow. And those are actually widely used right now. We posited, though, that by bringing in organoids, which have everything but flow, and combining that with this organoid on chip, these perfusible models, we could actually create a model that has a higher degree of physiological relevance. Now, the sacrifice in doing so is the throughput is much lower than cells on a dish or things in transwell membranes, or even organoids that can be high throughput, pick and placed into multi-well plates, right? So you're getting an enhancement in the physiological relevance, which I'll describe in, I think, some detail in the next several slides. But the sacrifice, of course, is throughput. So ultimately, as we think ahead, like how can we really take advantage of this, but also gain in throughput? And that's something we're thinking about. And I welcome suggestions from those of you in this space. So why work with kidney organoids? Well, first of all, if we look at the types of structures that are observed in these building blocks, we see glomerular-rich compartments. So just like these podocyte-rich clusters, like I showed you, that are responsible for the blood filtration. We see proximal tubule segments, distal tubule segments, and we even, in static culture, following like published protocols, we get endothelial progenitor cells. What happens, though, is if we keep these organoids in static culture, which is where they would be if they were in a 1536 well plate, high throughput screen, the endothelium retracts. So they're there, but they don't remain. And that's what I said. We posited that maybe based on some of our earlier proximal tubule on chip observations, that if we introduced flow, we could not only enhance maturation, but could we enhance vascularization? Could we sustain that vasculature for long periods of time? And the answer is yes. And let me just tell you a little bit about the chip that we made. This is a bio-printed gasket. It's just a silicone. It could have been molded. But we fill this with an extracellular matrix, and it's an adherent matrix, so the organoids actually stick and adhere to the matrix and spread out laterally. And it's made of gelatin and fibrin. And so we seed them here. This is a cross-sectional cut through here. You can see where they're placed in the chip. And then we subject them to flow. So we have an inlet and outlet. It's a pretty simple chip. And we're placing them onto the chip at day 11 in this protocol where they're in this pre-tubular aggregated state. So we basically have a metanephric mesenchyme. It hasn't been fully differentiated yet. But over time, as we continue to provide flow, we're continuing to mature and drive that differentiation. So if we look then at this series of images, and these are stains for CD146, which is an early marker for endothelium, and CD31, which is a later stage mature marker for endothelium. Podocalyxin is highlighting the glomerular podocytes. You can see that in a U well, if you just take these static organoids, do the differentiation protocol, there is some minor evidence of microvessels. Those do recede. In static conditions, with this ECM that we've developed, again, you can see a small amount of microvessels. But as we go to low and high flow conditions, we get a pronounced increase in this microvascular state. And we can see that between static and high flow conditions, even in the endothelial progenitor population, we have an upregulation of about 3-fold. So indeed, flow is making a huge difference. And you can see this very nicely with the fold change in all of the early stage markers of vascular cells and at the end with CD31. So flow makes a huge difference. And if we look at organoids that are adjacent to one another, we see that these microvessels that form actually connect. They form luminal connections to adjacent organoids. And they undergo angiogenesis. There's tip sprouting. And they're also recruiting mural cells, which line and support the microvessels. And that, we think, is important for sustaining them. Now, the next question, of course, to ask is, what are the impacts of this on the maturation of the tubular compartments and the glomerular compartments, and how are these microvessels interacting with those compartments? So first of all, we see, if we look at just do image analysis and look at the distance between the capillary and the proximal tubule, shown here in blue DAPI stain, only, we see that we get a big increase in the contact area. So the proximity of the vessels to the proximal tubules where reabsorption occurs increases. The separation distance concomitantly decreases. We also see that if we take one of these proximal tubules and take a slice through it, under high flow, we get a much more beautiful cuboidal-like cell morphology, and we also see ciliation. Okay, each one of these cells should have one primary cilia, here stained in red, and we see a pronounced increase in the ciliation of those cells. So flow is definitely enhancing the morphology of these cells. It's bringing vessels closer to the tubules, but we also see a concomitant upregulation of key transporters. So the transporters of glucose, for example, which are important to return nutrients back to the bloodstream. Aquaporin-1, which is involved in water transport for homeostasis. All of this is upregulating under flow. So the next thing we wanted to ask is this also happening in the glomerular compartment? And during development, what happens is that there's what's called S-shaped body formation. The nephrons sprout from the collecting duct, the ureter progenitor cells, and they form into the glomerular compartment, and they undergo what's called this S-shaped body formation. And we see in these organoids that evidence of that same kind of phenomena where they're recruiting a capillary that's going into this glomerular tuft and invading, and that's important, right? If we don't get blood vessels into the glomerular compartment, we can't do the filtration. So we can also see that the amount of invasion, again, PCAM here is the same as CD31, so we can see that the percent of invaded glomeruli goes up substantially in the presence of high flow versus static conditions. So we're getting a win-win-win. We're getting more vessels. The vessels are staying. They're not retracting. We're maturing the glomeruli. We're maturing the tubular compartments. And to just convince you that the glomeruli are not only getting invaded and vascularized, we also see an upregulation of the key maturity markers like nephron and synaptopodin. We also believe, and we posited that the increase in VEGF signaling that's coming endogenously from the podocytes is what's recruiting the vessels to come invade into the glomerular compartment. And I won't show you this data, but when we either suppress this endogenous VEGF production or we add exogenous VEGF, we can completely suppress the invasion so that that recruitment is not happening. Now, importantly, if we take a TEM image of these glomeruli, we actually see primitive foot processes developing as well. Now, this is a far cry from the beautiful podocyte foot processes that I showed in the human kidney, but the fact that we can see this at all is actually quite promising. Okay, so I'll leave you with that thought. Now, the next thing that we wanted to do was to use these organoid-on-chip models to study a class of molecules being developed at Roche, who funded this work, and it was actually a strong collaborator, known as T-cell bispecific antibodies. These were being developed for leukemia, and they were targeting WT1. And so what we could do on our chip model would be to seed the organoids again on chip as I described, provide superfusive flow over the organoids on chip, but now we could introduce both PBMCs, pure blood mononuclear cells, the immune cells, alongside these TCBs and look for, in this case, on target but off tumor effects, right? So the TCBs are being developed to attack a blood cancer, but they also attack any cell that expresses this WT1, right? So we wanted to understand if there were any off-target effects, and this is important because, as I'll show you on the next slide, podocytes express WT1. So they were very concerned about would there be damage to the kidney in this, on-target but off-tumor response. So the way they engineered this T-cell bispecific antibody was to bind to WT1 presenting cells. So they had these WT1 cells intercellularly create a peptide, HLA-A2, that presents itself on the cell surface with this RMF peptide. And the WT1 TCB binds to that, and it has a CD3 arm that binds to the T cell that recruits it to go ahead and kill that cell. So with that then, as I said, as I show here, the concern was podocytes also express WT1. So they wanted to use our model to make it an immune-infiltrated system to understand what was happening perhaps in vivo as patients were taking or being administered this TCB. So we had to do two things. We had to first convince ourselves that PBMCs could be circulated through our chip without damaging the blood cells themselves, without damaging the T cells or the B cells or any of these immune effector cells that are being circulated through the chip. And you can see that we have a high cell viability where the green percentage here is about 95% living cells over this 5-day period. So the PBMCs can survive being circulated through our chip over a 5-day period. But the second, of course, big thing was will our organoids, will the podocytes within the organoids actually present on their surface this HLA-A2 that the TCBs need to bind to? And in human adult kidneys, that's the case. But organoids, of course, are very immature analogs of kidney tissue. But as I'll show you here, under both static or flow conditions, we have roughly 50% of these cells exhibiting HLA. And if we stimulate these cells with interferon gamma, which upregulates HLA, we see a pronounced increase. So we use these without stimulation under flow, so not every cell presents, but it's a good enough analog for testing these TCBs. So we were provided from Roche 3 different T-cell bispecific antibodies. A positive control, which kills cells indiscriminately. It just binds to all kinds of cells, including the T cells, of course. That has the CD3 arm. The specific targeting WT1 TCB, which should only bind to cells like podocytes or the leukemia cells, and a negative control, which binds to the T cell but has no binding to any other cell, right? So we had the positive, a negative, and the sort of selective TCB. We introduced these two positive and negative controls on chip, and we looked at dose-dependent response. And we see that in the presence of the negative control, there's no change in the CD8 expansion. But in the presence of the positive control, we see over, as we increase the dose, this is day 5 on chip, that there's a pronounced increase in the expansion of CD8 positive cells. And if we look at then the killing of those, the CD8-induced killing using a LDH assay, over this 5-day period, again, this dose-dependent curve shows that the either no PBMCs or the negative control, there's really no change in either cytokine release, granzyme B release, or cell killing. But in the case of the positive control, as we go to higher doses, we get pronounced cell killing and pronounced granzyme B release and cytokine release. So they are indiscriminately killing lots of cells. So the real question is, what happens when we introduce the WT1 compound? And this is the one that targets only the WT1 expressing cells, the podocytes. And so they gave us a labeled version of this shown in white. WT1 is stained here in red. Because of the DAPI stain, it looks kind of magenta. You can see that throughout the podocyte risk cluster, we have WT1 targeting those cells. And over time, we see a pronounced degradation, if you will, in these glomerular-rich features. And we can see that with a WT1, we get a really high amount of cell killing for the WT1 star TCB. If you're sharp, though, you're also going to see that we have some dead proximal tubule cells. So LTL positive is the marker for proximal tubule cells. We really wouldn't expect proximal tubule cells to be killed by WT1, and yet some cell killing is occurring. So we went in and we looked very closely at this interface between the glomerular compartments and the proximal tubules, which of course they connect in vivo as well as in our organoids. And we see that there is a region here where there's a transition zone where some LTL positive cells actually also exhibit WT1. So there's a transition zone between the glomerular podocyte cells and the LTL, pure LTL proximal tubule cells, where they're expressing both LTL and WT1. And it's these cells that are the fraction of the LTL positive cells that are being killed, not the entire LTL rich population. So that actually did end up making sense. And that's shown here. But also, if you're thinking about the way our model has been developed, we're flowing the media and the TCBs and the PBMCs over the surface of our organoids. That's not really how we should be physiologically delivering the TCB or the blood cells, the immune cells, to these organoids. Ideally, we would like a perfusable organoid system, okay? So even though this was kind of an interesting result, the caveat is that maybe in reality, this TCB would never get through the glomerular basement if it was delivered systemically through the blood network, right? The blood vasculature. So that led us to do the next thing, which was to try to create a perfusable kidney organoid on chip. This is relatively recent work. We have not yet introduced the TCBs to this organoid on chip model, but I think I can convince you that we now have a route to do so. And so let me tell you just a little bit about this chip design. It's designed so that we have two vessels, a lower channel that is going to be seeded with endothelium, and an upper channel that we're going to seed with organoids. And you can see that here in this bright field image. Lots of organoids are kind of sitting in this channel. The entire channel diameter is here. The entire chip is embedded again in the ECM. We have flow coming in and out. This is the macro vessel. And what we're going to do is use flow, superfusive flow in this channel to upregulate the endothelium. We're going to introduce a chemoattractant through the endothelialized channel, and we're going to coax the microvessels to migrate to the macrovessel and connect. Well, at least that was the goal when we designed the chip. And it turns out that it actually worked. So let me tell you a little bit about that. So first of all, this is a top-down view of kidney organoids that are seated in that top chip. Again, just getting superfusive flow initially. So we're flowing independently through this channel initially, and in the bottom channel, which we seed with endothelial cells. You can see, again, the upregulation of the endothelium. We see a nice, pronounced macrovascular network. We can see podocyte-rich clusters, et cetera, et cetera. If we seed the bottom channel, in this case, we seeded them with HUVECs and not iPSC-derived endothelial cells. But you can see that the cross-section of that channel has a nice confluent endothelium. And through a barrier test, we can show that in the absence of the endothelium, we get pronounced leakage of dyes that diffuse into the surrounding extracellular matrix. But when we have that confluent endothelial barrier, that's greatly suppressed, particularly at the highest molecular weights of dye that we're perfusing through. So now we have the ability to perfuse through the bottom channel, and if we can connect the microvessels to the organoids at the top, go through those and then out the top channel, right? So the question is, how can we drive anastomosis? So on day one, with these organoids in this top channel and the endothelium in the bottom channel, you can see it, I think, much more clearly in these confocal cross-sectional images. You can see that over time, perhaps you can see it, there's a front that develops where the cells are migrating from the top channel down to the bottom channel and connecting to the macrovessels. And you can see some tip sprouting here also from the macrovessels up as these cells are migrating down. So the endothelium cells present in the organoids are migrating down to the macrovessel. Tips are sprouting from the macrovessel. And the question is, do they form anastomosis? Do they connect? And indeed they do. And this is maybe perhaps a much more clear image of what I just showed you. Again, where now the endothelial cells are stained in CD31 and shown in white instead of red. You can really see quite nicely that these microvessels are sprouting, and these microvessels are migrating, and these macrovessels are tip sprouting. So we asked ourselves, like, do we have lumen-on-lumen connections? And this is just one 3D reconstruction. If you look here very nicely, I think you're going to see this vessel opening up and continuing to go up. So all of this is luminized. And this is the printed macrovessel. And these vessels here are coming from the top channel that has the organoids. I think you'll see this in a more convincing way on this slide, where the red stained vessels are from the macro vessel. So they don't have GFP. They haven't been labeled with GFP. They're just stained in red. And the green vessels are GFP labeled. And you see these interconnects here in yellow. That shows that the red and green are connecting, right? So we get luminal sprouting from the macro vessels. We get the microvessels coming down and migrating. And some distance away from the macrovessel channel, if I just go back, some slight distance away is where the anastomosis occurs, where these macro and micro hookup occurs. So the question then is, can we deliver things like red blood cells through the chip, through these interconnected now anastomosed capillaries to the organoids themselves? And this is just an image that shows red blood cells sitting in these capillaries. Now we're zoomed in on the organoids in the upper channel. You can see some capillary invasion. This one is not invading, but you can see red blood cells are being able to be delivered through the bottom macro vessel, hooked up to the microvessels and going up into the organoids themselves. So now we have a way to actually do the experiment that I showed you with the TCBs in a little bit more proper format. Now this is actually important also, this ability to have macro-micro integration, macro vessel integration is also very important when we start thinking about scaling up from just single organoids on a chip to taking these as building blocks and building vascularized tissues. This is work that's very much in progress. It builds on some work that came out of our lab a few years ago, and I'll describe that. But we're taking baby steps, is what I would say. And I'll show you some of our earliest experiments with implanting these tissues in small animal models. So let me tell you, I think I've already explained why this is an important problem, right? End-stage renal disease, you know, like 1 in 7 people have chronic kidney disease, and it progresses over time from stage 1 to stage 5. When you get to stage 5, you have to go on dialysis. My uncle currently is in end-stage renal failure. And so it has a personal tie to me, but it's also something where I think there's a real clinical need. I've talked about the fact that, you know, the wait times are typically on the order of 3 to 5 years. And importantly, every day, of the order of 13 to 17 people die waiting for a kidney transplant. So we're not meeting the needs. There is some really good work being done with xenotransplantation, eGenesis, for example. I don't by any means want to say that this approach is the only way we're going to solve the problem. But I think in the end, if we think about whether we want donor organs from a humanized pig or we want to ultimately be able to construct organs that are from our cells for us, I think you can see like where the future might end up. And so we're trying to establish some foundation to get to that path. So I've told you about kidney organoid building blocks, about why I think they're really the right building blocks to build tissues, just like we think as material scientists about what are the right atoms or molecules to build materials. I think these building blocks are important rather than just using single cells, because you would have to have a printer that had the precision of placing every specific cell type where you wanted it. And to build a tissue the size of my fist, which is what our kidneys are roughly in volume, that would take months or years. So that's not feasible because cells only live for about 1 hour while you're trying to print the tissue of interest. So the less you can print, the better. I've showed you that there's promise for if we can embed vascular networks inside tissues to connect to the microvessels that we know are present in the organoids. And so the last piece I want to describe in the rest of my talk is this process known as SWIFT, sacrificial writing in functional tissue, which allows us to take top-down printing and embed larger channels inside densely cellular matrices that are composed of these building blocks. So I don't think I have to convince this audience, since it's a fairly bio-focused talk, but if we don't have blood vessels within about a few hundred microns of our cells, the cells will die. So if we take embryoid building blocks, which are basically our human-induced pluripotent cells, and we aggregate them into sort of objects that are, say, 100 microns to 500 microns in diameter. They're granular in scale. So if we think about some colloid science, these are much, much bigger than colloids, so they're really just granules, if you will. If we take these building blocks and we make a tissue that's 4 by 4 by 4 millimeters on a side, a cube, and we're using these granular building blocks that are containing these densely packed living cells, our tissue cell density is approaching what you would find in our organ. That's organs level density, 200 million cells per milliliter. If we take that cube and we place it in media, so the outer periphery is getting oxygen, it's getting nutrients, we can see very quickly that the inner core is all necrotic. These cells are dead. Oxygen diffusion can only sustain cells for the first few hundred microns, and we have a core that's essentially not living. And this is after only 12 hours in culture. So this motivated us to develop this method where we could take any kind of organ building block. We've done this with cardiac organ building blocks. I'm going to focus today on kidney organ building blocks. You can do it with cerebral, not that we've focused on that, but you can. Pretty much any organ building block. It can be an organoid, or it could be a multicellular aggregate, a spheroid. It doesn't have to be derived in an organoid form, but something that's granular, multicellular, that can be compacted to form a dense matrix that's living. So we suspend these organoids in an extracellular matrix that's fluid at low temperatures, and then as we warm the tissue, that matrix basically forms a glue. And that allows us to write this sacrificial ink. Let me start playing this video. Write a sacrificial ink inside this densely cellular matrix. So what you're seeing here is a sample that contains 2.5 milliliters of embryonic bodies, so undifferentiated stem cell aggregates. It contains about 500 million cells at that cell density that I described. Everywhere you see a red feature that's being printed, that will ultimately become an open channel, an open lumen. And again, you can see an inlet and outlet, so we can perfuse this tissue through this vascular network. So we call this a sacrificial ink, and because it's being written in a functional tissue, Mark Schuyler Scott and Seb Buzel, who are the two postdocs that carried out this work, named the process SWIFT. And Mark is now at Stanford as an assistant professor. And I had to give a shout out to him because he was one of the first 8 awardees of ARPA-H funding to build a whole human heart using this kind of technology. So yeah, pretty exciting, 26-some million dollars, and he's off and running, so I'm very proud of him. Now, if we look at this, you need a lot of cells and you need a lot of organoids or building blocks to build a tissue. And we also wanted to make sure that we had the appropriate rheology to allow us to write these features in the tissue. They would stay where they were, where we wrote them, and they could be erasable. And so we developed the sacrificial ink and the matrix, the living matrix, the embryonic body matrix or organoid matrix. You can substitute those interchangeably. And we showed that they're both shear thinning and that they're viscoelastic, meaning that they have a solid-like response when they're not being perturbed. But if you go above the shear yield stress, they flow. Right? So both the ink will flow as you're writing, but the matrix, the living matrix, will also flow. And you can see this very nicely in this video, where what you see are the embryonic bodies being very densely packed. Here's a nozzle coming through, translating. Nothing is being printed right now. I just want to show you that the matrix can self-heal. It can flow around the nozzle. Now, the next pass, we're actually depositing the sacrificial ink, and you can see that these embryonic bodies are kind of being pushed away. Now, the trick is at low temperatures, 2 degrees C, as we're moving the nozzle through, the ECM that's essentially coating each of the embryonic bodies that are in a solution is fluid. But when we warm the tissue up to 37 degrees C, that basically solidifies and locks all the organoids in place. And at the same time, concomitantly, the solid-like ink at low temperatures becomes a liquid, and we can flush it out of the channels. So I just said all of this in words, but let me show you what happens, just maybe focusing on this data alone. As we increase temperature, the sacrificial ink melts and fluidizes, so we can remove it to get these open lumens. And the extracellular matrix starts at low temperature as being fluid-like, and then it solidifies and forms a glue. And you can see the extracellular matrix here labeled in collagen 1 stain, and it basically glues all of those embryonic bodies or organoids together if we use differentiated aggregates together to allow us to remove the ink without having them collapse into the channel. If we didn't have this thermorheology, And you saw how easily the organoids moved as the nozzle translated. And we tried to take the ink away, they would all just collapse, right? And the lumens wouldn't stay in place. But they do stay in place, and we can change the size of the lumens simply by changing the print speed for a given nozzle diameter. You can see the tissue cross-section very nicely here stained. And they make a huge difference. Remember, there's that big red necrotic core without the printed vessels. Now we have, largely, most of the cells are living in this tissue cross-section. So the next thing we had to do was do this rather than doing it with embryoid bodies, like differentiating large volumes of kidney organoids, right? And so rather than just doing it in plates and in microwells, we established bioreactor protocols that would allow us to make large volumes of organoids for tissue printing. And yet they still maintain the cell types of interest, the podocyte-rich clusters, the LTL-rich proximal tubules, and the CDH1 distal tubules shown here. And they have evidence of the vascular components, CD31. So this is what they look like in bright field. This is what they look like in immunofluorescence confocal. And for this kind of reactor size, which is about 60 milliliters, we can get about 3 milliliters of densely consolidated living matrix. So we're going to print channels in here. And this one, I'm just going to show a very simple channel. We've made a bioreactor. This was Seb Uzel. It's coming down with a needle, a nozzle that's coming here, connecting to an inlet here, and just printing a single cylindrical vessel. So this is not very complicated vasculature, but we wanted to just, in the beginning, just understand, like, if we flow through the inner lumen, we also have flow around the tissue periphery. What happens to, do we vascularize the tissue? Do we see the kinds of things that I showed you on chip? Is that happening? You can see each of these samples in our incubator. They're being perfused, independently addressed inner and outer regions of the tissue. And I showed this earlier, but this is what, if we then come through and take a cross-section through the tissue, we can see on day one evidence of the boundaries of these organoids, right? The tissue still has some regions that are less dense and regions where you can see the individual features of the organoids themselves. But by day 7, the tissue has completely coalesced. And if we take a cross-section now, first of all, you can see that that lumen no longer is perfectly cylindrical because there has been some consolidation, right? Again, material science, we would call this like sintering, some sort of fusion of and consolidation. All that empty space here between the organoids is gone. The lumen is no longer perfectly cylindrical, but it can still sustain flow. And if we look then at the tissue cross-section and we zoom in to a region, again, we see microvessels, they're pervasive. We see glomeruli, we see our LTL-rich tubular compartments. And so we have a tissue now that's a bulk tissue. We see evidence of the same kinds of things that I showed previously, like invasion and wrapping of the blood vessels. We can go in and we can count the number of cells per unit area in the tissue and so forth. So the places that we are now is trying to basically print a human kidney that's mouse-sized. So a mouse-sized kidney is about if you look at your pinky finger and you look at your fingernail, that's about what we're trying to print, right? It's not very big. And this is the printing process going on. And let me show you what we're actually printing. So this is the schematic of this. We have a simple single vessel. We have a little drainage channel here to try to take the filtrate out. We've designed and built this pedestal. It's a suturable cuff. We can cut through this. There's just a base to connect to the inlet and outlet pins for in vitro perfusion. But when we cut through this, we can suture the outer two legs to the vein and artery of the animal, and the inner one to the ureter for filtrate removal. And if you can imagine, the size of this is actually tiny, tiny. If I had a quarter, it'd be sitting, kind of in the middle of a quarter. But once we do this, we remove the extra loaded living matrix, so we don't need all of this. And we have this tissue, basically mesh, that we suture together here to form the final encapsulated structure. So we have the cuff, which we cut here prior to implantation. We have a suturable cuff that basically encapsulates the living matrix and the channels that we've printed inside. And we're working with Leo Raela's lab at MGH now to implant these tissues into mouse models, humanized mouse models. And this is just one sequence where we've made 4 tissues. You can see the sutures here, just keeping the tissue mesh together. It's a clamshell design, so initially when we're printing, the mesh is open, but then after printing is completed, we close it. This helps with handling as we bring the tissue into this cavity. So we're really doing a complete single nephrectomy. We're taking out a mouse kidney and we're replacing it with this Swift constructed mini human mouse-sized kidney. And you can see that we have inflow and outflow. We're suturing to the artery and the vein. And it works, okay? We've sustained this for periods of up to an hour. It's not a lot, I can tell you that. We have to go much, much farther. So we can laugh at that or we can cheer at that. I'm not sure. It's a little of both. We're cheering because we had, up until very recently, much lower time. But one of the things that we're continuing to work on now is when we do the anastomosis, this tissue is still pretty fragile, and we didn't endothelialize those channels in this case. And there are reasons for that. We thought we could try maybe just having the native endothelial cells vascularize the internal channels. But we found pretty quickly, and maybe it's intuitive for those of you who have done these kinds of animal surgeries, that the burst pressure, that when we do the anastomosis, oftentimes we find that we were getting some tissue rupture. So we're kind of back to the drawing board a little bit here, but where we're going is we're now embedding a more biomimetic endothelium inside these tissue architectures using what we call core shell printing. So not only are we going to endothelialize the vessels that we embed into the tissue, but we're also going to have smooth muscle cell wrapping to create a more robust vasculature. And we've de-risked this in acellular matrices as well as in cardiac tissue matrices. So we're now porting this over to our kidney tissue work. And the other thing that you might have been thinking throughout this whole presentation is, great, you have all this nephrons, and you're thinking you're going to make filtrate, but what about the collecting system? Do you have that, right? And so we're also simultaneously starting to work on stem cell-derived approaches to creating the ureteric buds, which then form the collecting duct networks, and trying to bring those into the tissue as well. So I say baby steps. I think we've just de-risked quite a bit, frankly, but we also have a huge way to go. All right, so I'm giving you a look into some of our unpublished work and where we're really going. But something I'm very excited about for our team. So with that, I'll end. I will thank the many folks that fund us, the broader lab, which includes both the materials component as well as the bio work that we're doing. I'm just very fortunate to work with a number of amazing people, both former postdocs like Kim Homan, who led all of our vascularized organoid on chip work, and Katharina Kroll, who was a PhD student who kind of followed on and did the TCB work that I described and the macro vessel hookup work. Jonathan and Kayla are doing the UB organoids, and Seb Uzel, who's still in our lab, who was like a co-pioneer of Swift, is just one of the most amazing engineers I've ever had the privilege of working with, and he's an MIT PhD from Roger Kam's lab. And we've been very fortunate to have a number of incredibly fruitful collaborations. So with that, I'll just end and thank you all, and take any questions.
We have to raise the lights for questions since
Okay,
we
fair.
have,
Fair.
but questions.
Yeah, Maggie.
Great talk, Jennifer. I was really impressed. Always, there are some pictures that I've seen again and again. I'm still very impressed by it. I have two questions. First one is regarding the media that you're using in these different projects. When you're adding T cells or when you're adding a separate channel of HUVEC, et cetera, what is your philosophy about the compatibility of these different media that you're using?
Yeah. So we definitely have a more kidney-centric media in the top channel that we're superfusing over the organoids, and a RPMI media for the endothelial cells. And so, yeah, that could be an issue for some of these chip designs for sure, but that's how we have handled it. Yeah.
Gotcha. And the second question is regarding the vascularization part. I assume that in the kidney, there's probably macro vessels as well as micro vessels. So there should be a distribution of the blood vessel sizes.
Correct.
And I wonder how this is taken care of in the kidney organoid chip work that you've been doing, or do you think that having different sizes is something that's important, or as long as we have microvasculature that is accounting for it is sufficient?
No, that's a great question. I think what I tried to show you is that we can print a myriad of channel sizes from over a 10x range. We don't have control over the microvessel distribution. That is coming endogenously through the differentiation protocol and flow. So we don't have control at that fine scale. But certainly, as we think about scaling this up beyond a mouse-sized kidney, we're going to have to be able to print and embed a hierarchical network of vessels. And some of the work that I didn't get a chance to show you from Paul Stankey, at the end I hinted to this coarse shell printing of the biomimetic vessels. We're absolutely following Murray's law, doing the kind of biomimetic branching that you would have in vivo. So yeah, I think that's actually a pretty solvable problem. I think one thing that I also glossed over that is a less solvable problem. It's all about plumbing in the kidney. So it's great that we can get these vessels, and I think have established pathways to link them up. We've shown that we can get invasion into the glomerular compartments. We haven't yet, in any convincing way, shown that we can get filtrate production. Okay, and even if we do in the glomerular compartment, we have to get that filtrate out. So just like we have to embed this pervasive vascular network, we have to embed a pervasive, drainage network, which is kind of where we're going. We're learning as we go. We think it's important to kind of keep pushing on the in vivo experiments, because we also know that there will be, based on just individual organoids that have been implanted in the renal kidney capsule, we know that in vivo there will be vascular invasion and also continued maturation. So we want to always know what can we expect biology to do in vivo? How much more maturation and function will we get versus what we're engineering in vitro, right? We don't know the answer to that yet, because we haven't, as I told you, been able to do long duration implants in the single nephrectomy model. We can put in discs, we can put in things like that, but really where we get perfusion and anastomosis, that hasn't been done by anyone, not us, not anyone. But we think those experiments are important, even if we're only halfway there on the nephron side, and we still have more to do on the collecting duct side. But we're working in parallel, and so that's where we're going. It's a good question.
Thank you, Jennifer. That was really exciting. I was wondering if you may have already explored this space, but looking at a tumor and the relatively different vasculature of a tumor, is this something that your lab has looked at, and is there a way to examine what the impact of flow is on the permeability of vasculature in tumors?
That's a great question. I mean, we have recently reported a tumor on chip model, but we haven't yet done the vascularization piece for that model. We have a permissive microporous matrix that we're introducing T cells into, and we're looking at interactions with the tumor model. But the next ongoing work is the vascular network. So, I mean, I'd love to talk more with you about that, especially if there's ways to collaborate.
Oh,
Yeah.
that would be really exciting. And the blood-brain barrier, you had mentioned that's something that your lab has also been thinking about?
Yeah, we have been. And so the strategy on the cerebral organoid side is very different, because we don't get an endogenous population of endothelial progenitor cells when you go, because they're, different germline. So mesoderm can get you kidney and vessels, but not for, the cerebral. So there is where we were working closely with George Church's lab to use ETV2 DOX-inducible cells, where we can sort of a priori say we want this ratio of neurons and this ratio of endothelium. And that, I think, is a probably winning strategy for starting to think about blood-brain barrier. Yeah. There's a lot to do. We decided to do the hardest possible organ on the most far-reaching thing, as opposed to all the cool stuff that, well, we're doing a little cool stuff along the way, but I don't know, we're locked and loaded on this bigger thing. and there's lower hanging fruit, but anyways.
Thank you for that really nice talk. I'm really curious, given you the challenge of then adding in the filtrate network, can you take the same approach?
Yeah.
And VEGF is a very powerful vascularizing growth factor. Is there something similar you can do for making the filtrate network, or do you have to take a different approach?
No, there is. And we're part of this NIDDK Rebuilding a Kidney Consortium, and there are others that are identifying the factors that will kind of drive that hookup. And so we're taking those learnings, and we're definitely going to use that same thing. We're going to have a drainage channel. We're going to try to connect to the distal tubules. Right now, if you take a single organoid, because it has a periphery and it's not connected, I mean, there's blind ends on all of those tubules. Like, they come out of the glomeruli, but they dead end, basically. And we're working with Lisa Satlin's lab at Mount Sinai in New York, and they're amazing. They can take these little tubules, one tubule out of our organoid, and they do this for mouse kidneys as well, and microperfuse them, right? It's a pretty cool stuff. So we know that they're functional. We absolutely know that. We just need to get the plumbing. It's a plumbing problem. It absolutely is.
Thank you.
Yeah.
Hi.
Hi.
You're kind of touching on this already, but I'm wondering if you have any idea of, you engineer the tissue to the best of your ability and it is producing filtrate, what kind of efficiency it has. So, a real native organ has all the microstructure in the right place, but you're starting with these organoids.
Yeah. So I think you've already asked a really good question, so I'll jump in. First of all, we know we can scalably generate the same, the number of nephrons we would need to have a therapeutic value. How we're thinking about it initially is that we don't need 1 million nephrons, we need maybe 200,000. We need to restore 10 to 15% of glomerular filtration function to move somebody from stage 5 to stage 4 dialysis, stage 4 kidney disease. That just takes them off dialysis. That's enough, actually. We don't have to replace their whole organ. Okay, just to say that to begin with, right? That's the ultimate goal, but that's, an intermediate step. So your question is, what amount of filtrate can we produce? So, there are protocols that will allow us to basically inject something like FITC inulin and watch clearance, and we can measure that in vivo, right? So we're not there yet, but that's where we're going. And then we'll know how many nephrons do we need in this tissue to get 10 to 15% glomerular function restored. Boom, that's it. Yeah.
Thank you.
Last question.
Jennifer, elegant talk, really nice work. It's amazing to see the bottom-up assembly coming along so nicely. My question is really, you mentioned the top biomanufacturing.
Yeah.
How do you think about the challenges in scaling this in a different manner, right? Not just now scaling from the
Yes.
assembly, but to
Yes.
the tens of millions of patients globally that might need these treatments.
So this is the part of the work that I haven't described, and the question is, I love the vision of made for you, made from you for you. I love the vision of taking individual patient cells. That would require quite a lot of machinery to have individual banks of cells per patient, organs derived developed and built for patients. So the other thing that's, of course, happening is, and we're making hypoimmunogenic organoids in our lab too, trying to take down the immune system in the cells that we're putting in, right? So making them, removing and controlling the HLA mismatches to minimize rejection. Hypoimmunogenic cell lines probably won't get you all the way there. You're probably still going to have to be on immunosuppressants to some extent, but there is a path forward if you wanted to have like a simpler cell factory, if you will. There is a path forward. I'm still like kind of really excited about the personalized one though. But it probably isn't going to happen. That won't be the first thing out of the gate. That probably just isn't feasible from a cost perspective. Yeah.
I have one final task. I said in my opening remarks that the lectureship, the Michaels Lecture started in 1995. That means I am presenting you with a plaque in honor of the 30th annual Alan S. Michaels Distinguished Lectureship. Thank you so much for an invigorating talk with a lot of hope for the future.
I hope. Thank you so much.
Eyes are open. You guys look great. Thank you so much.
Thanks.
Nice.